From a genome to function, selection and isolation of functional genes. Imagine that a cell is like a vast library containing thousands of books. Each book represents a gene, but not every book is being read at the same time. Scientists are often interested in finding a particular gene that performs a useful function, such as producing insulin, conferring disease resistance in plants, or helping microorganisms degrade pollutants. The process of identifying and obtaining such a gene is known as selection and isolation of functional genes. So, what is a functional gene? A functional gene is a DNA sequence that is actively expressed and produces a biologically important product, usually a protein or functional RNA. Like, for example, insulin gene in humans, nitrogen fixation genes in bacteria, disease resistant genes in plants, antibiotic resistance, no, I mean to say antibiotic producing genes in microorganisms. So the first challenge is identifying which genes among thousands is responsible for the desired function. And next is the selection of the genes. Selection refers to identifying the gene of interest based on its known or suspected function. Selection based on phenotype. Here, scientists observe a specific characteristic and search for the gene responsible. For example, a plant shows resistance to fungal disease, and researchers compare resistant and susceptible plants to identify the resistance gene. And selection is also based on protein product. If protein produced by a gene is known, scientists can use antibodies or protein sequencing to trace back to corresponding gene. Like, for example, isolation of human insulin gene was facilitated by knowledge of the insulin protein sequence. Next is the selection using molecular markers. DNA markers linked to a trait help locate the desired gene. Example, RFLP markers, SSR markers, SNP markers. These markers act like signposts guiding researchers to the gene. Differential gene expression studies like involve genes active in one condition but not another can be identified using RNA sequencing, microarrays, RT-PCR. For example, genes activated during drought stress may be selected for developing drought-resistant crops. Isolation of the functional gene is like once the gene is selected, it must be isolated from the genome. And here, genome isolation is involved, and where DNA is extracted using basically cell lysis, removal of protein and contaminants, and DNA precipitation, followed by purification of the DNA, which serves as the source of the target gene. And that is then followed by PCR amplification. PCR is commonly used to isolate a specific gene. Here, what happens is we design specific primers to the gene and then amplify the target DNA and verify the products using the agar gel electrophoresis. And the PCR allows millions of copies of desired gene to be generated within hours. And next method is cDNA isolation. For genes that are actively expressed, first the mRNA is isolated and that convert those mRNA into complementary DNA using reverse transcriptase and amplify that cDNA. So the advantage here is cDNA contains only coding sequences and lacks introns. And next method is DNA library screening. Here scientists create genomic libraries and cDNA libraries, and these libraries contain thousands of DNA fragments. The desired gene is identified using DNA probes, hybridization techniques, colony screening, and next one is the verification of the gene function. So after isolation, scientists must confirm that the gene truly performs the expected function. So the methods include gene expression analysis, RT-PCR, real-time PCR, northern blotting. These determine whether the gene is actively transcribed, and protein detection is by Western blotting, ELISA, and this confirm protein production. Functional assays involve the isolated gene is introduced into another organism. For example, like a bacterial cell receiving the insulin gene begins producing insulin, confirming gene functionality. And then the cloning of the functional gene. The gene that is isolated is inserted into a vector such as plasmid. Here, the gene and the plasmid are cut using specific restriction enzymes, and the DNA ligase joins the fragment, and the recombinant plasmid which is introduced into the host cells. And the host cell multiplies, producing many copies of the gene. This process is known as the gene cloning. So the application of the functional genes involve functional gene isolation include production of insulin, growth hormone synthesis, and even in gene therapy. In agriculture, it involves pest-resistant crops, drought-tolerant varieties, improved nutritional quality, and in industry includes the uses such as for enzyme production, biofuel generation, bioremediation, and in research, it includes the understanding of gene regulation, studying diseases, and developing novel therapeutics. So, the selection and isolation of functional genes from the foundation of modern molecular biology and genetic engineering, and by identifying a useful gene isolating it from the genome, verifying its function, and cloning it for further study and application, scientists can harness nature's genetic information to improve medicine, agriculture, industry, and our understanding of life itself. So, the key message for the students is that every remarkable biological trait from disease resistance to insulin production begins with a functional gene. So learn to how to select and isolate these genes in the first step toward unlocking the potential of and all the power of biotechnology. Thank you very much.